Professor Ben-Ari is amongst the 5 most quoted French neuroscientists. His discoveries on brain development in health and disease have been recognized by major awards. After 4 decades academic research, Professor Ben-Ari shifted to therapeutic research, founding and directing NeuroClore, a startup dedicated to treat brain tumors.
Brain tumors, and in particular glioblastoma, are pharmacoresistant disorders for which there is no significant improvement in spite of huge efforts. My aims are to develop a novel approach to treat brain cancer and in particular glioblastoma.
NKCC1 is present on the leading edge of migrating cancer cells, controlling migration and metastasis. In addition, glioblastoma grade severity is directly related to NKCC1 hyperactivity. Blocking NKCC1 by drugs like Bumetanide or by NKCC1 KO attenuate the severity of tumors and enhance survival in animal models. Parallel experimental observations suggest that the anti-parasitic drug Mebendazole that inhibits microtubule formation also induces apoptosis and cell death. In a U.S. Phase 1 trial, Mebendazole administered for several months with temozolomide, an anti-neoplastic agent, prolonged survival.
We reasoned that combining 2 different drugs with different targets and different mechanisms of action might lead to complementary and possibly synergistic effects on brain tumors and notably glioblastoma. In this aim, we used human cell lines of glioblastoma and freshly removed tumoroids to test the effect of these 2 agents separately or together. We confirmed that they have in certain cases a complementary and even synergistic action. Relying on this, we have performed 1 pilot trial in a person with a very severe metastasis in the brainstem, a huge one. And indeed, we increased her survival time and reduced dramatically her sequels for 10 to 11 months in conditions that were quite unexpected considering the severity of the tumor she had. We are now working on the possibility of extending this and making more pilot trials in more patients having very different types of brain tumor.
In parallel, working with a leading chemical company and pharmaceutical experts, over 120 novel NKCC1 inhibitors have been synthesized and patent protected, most being between 75% and 120% of Bumetanide NKCC1 inhibitor efficiency. They inhibit migration and trigger apoptosis in glioblastoma multiform cell lines and resected tumoroids. In rodent cortical brain slices, they enhance GABAergic inhibition and block seizures.
We anticipate that a novel molecule-protected agent, newly synthesized, will constitute a major asset to treat brain tumor. We trust that a new molecule inhibiting NKCC1 will be efficient to treat a wide range of other disorders, a sort of aspirin of brain disorders at the long run.